I'll be presenting on some common pesticides and their alternatives in agriculture. So pesticides in general are just substances that function to prevent, destroy, repel, or otherwise mitigate some type of pest. They are generally a chemical or biological agent, and they are most commonly used in agriculture to protect crops from pests such as insects, weeds, and bacterial or fungal diseases, as well as protect them during their storage and transportation from pests such as rats, mice, insects, or other biological contaminants. And they also can function to control vectors of human or animal diseases. An example of this would be the malaria vector. So pesticides can be classified based on their target organism. So insecticides obviously function to control insects, either by killing them or disrupting their growth and development. Herbicides kill or inhibit the growth of unwanted plants. These are known as weeds. So 80% of pesticides are herbicides. Fungicides will obviously work to control fungal problems like molds or mildew. Rodenticides kill rodents like mice and rats, and so on. So they can also be classified based on their chemical composition. So organochlorines, organophosphorus, and carbamates are three groups used as insecticides, and we are going to focus mainly on those. So organochlorines were the first synthetic organic pesticides to be used in agriculture and public health. They are hydrocarbon compounds that contain at least 5 chlorine atoms, and they are nervous system disruptors, and so they work to disrupt the sodium-potassium balance of the nerve fiber, and this forces it to transmit continuously. And this will then cause convulsions and twitching and paralysis of the insect that can lead to its death. They exist in varying toxicities, but they are resistant to most chemical and microbial degradations, and so because of this, they have a tendency to bioaccumulate, and so they have kind of been phased out because of that. DDT is the most notable example of an organochlorine. It was one of the first used pesticides. It was used during World War II originally to try and fight malaria. It did work initially, but there were several associated negative things like toxicities to humans and the environment, and so they have since kind of been banned in several places. So organophosphorus compounds have kind of have replaced them. They are toxic to both vertebrates and invertebrates, and they work to inhibit the enzyme acetylcholinesterase. We'll talk more about that in a bit, but this causes the acetylcholine to transfer the nerve impulses kind of indefinitely, and this causes a variety of symptoms such as weakness or paralysis. And unlike the organochlorines, organophosphorus compounds are more easily decomposed in the environment by the various chemical and biological reactions, and so they do not have as great of a tendency to bioaccumulate, and so that's why they're more acceptable to use. So again, they inhibit acetylcholinesterase. So acetylcholine is just a physiologically important neurotransmitting agent that is involved in transmitting the nerve impulses to the effector cells at cholinergic, synaptic, and neuromuscular junctions. It is found in a wide range of animal tissues, including mammals, birds, fish, reptiles, and insects. And so acetylcholinesterase regulates this activity of acetylcholine by reducing its concentration as it hydrolyzes it into choline and acetic acid. This mechanism is shown to the right, and this is mainly done by the acetylation of a serine hydroxyl in the steric site of the enzyme, and this is catalyzed by basic imidazole moiety B, which is histidine. The enzyme then rapidly breaks down to acetic acid in the regenerated enzyme and is deacetylated. So the mechanism and inhibition of acetylcholinesterase by organophosphorus is shown here. It's pretty much a two-step process. The serine hydroxyl moiety in the enzyme's active site is phosphorylated, similarly to how it is acetylated, but unlike the acetylated enzyme, the phosphorylated enzyme is more stable and is in some cases, depending on what R groups are attached, it will be irreversibly inhibited. And so, yeah, when the serine hydroxyl group is blocked by the phosphoryl moiety, and it is no longer able to participate in hydrolyzing acetylcholine, and as acetylcholine cannot be broken down, this will lead to the continuous stimulation of the muscle or nerve fiber. So carbamates are similar to organophosphorus insecticides. They have a high toxicity to vertebrates and invertebrates, and they also inhibit enzyme acetylcholinesterase. And again, like organophosphorus compounds, they are also more easily decomposed in the environment, and so they will not have as great of a tendency to bioaccumulate. The mechanism that they inhibit acetylcholinesterase is similar to pretty much identical to how the organophosphorus insecticides do this. The two-step process, kind of the formation of the enzyme inhibitor complex, then the carbamylation of the serine hydroxyl, and the carbamylation of the serine hydroxyl is how results in the inhibition of the enzyme. So again, pesticides, there are several benefits to them, but there are also some negative effects. There are some negative health effects, and there are also some negative environmental effects. A lot of the pesticides will contain some high organic solvent contents that are bad for the environment. Additionally, most of a lot of the sprayed insecticides, 98% of them and 95% of sprayed herbicides, they will reach a destination other than their target, and so this will result in pollution to the water and the soil, and other things that they were not intended. Additionally, there is poisoning of non-target species, as these pesticides are more broad spectrum and will react with what they are not intended to, and this can lead to a decline in pollinators such as bees. And so there are some alternatives to pesticides. So these semiochemicals are a type of biopesticide. They mimic some insect pheromones that relays the messages between the species. So the biopesticides will just trap or interfere with the reduction or reproduction of their target organism, as opposed to the conventional other pesticides that inactivate or kill them. As these semiochemicals are more pest selective, they will not negatively impact other organisms, and so this is better overall for the environment and human health. The disrupting of the mating pattern is achieved by just applying the biopesticide to the fields, and this can confuse the mating pest by overloading their sensory receptors. So some downsides to the semiochemicals is that many of their pheromone preparations that were done were accomplished by classic organic preparation techniques that were not very environmentally friendly, but more recently there's been considerable effort to figure out and implement more cost-effective chemical processes to achieve more economically more profitable, environmentally just better procedures. And so new techniques, one of these new techniques is types of olefin metathesis reactions, and these are better for the environment and more cost-effective used in green chemistry. And two examples of these synthesis of types of some pheromones are shown here through olefin metathesis. The second alternatives are these nano-based particles. So they are polymers that respond to changes in the environment, and the pesticides may be loaded into them, and this just allows for targeted delivery and the controlled release of nanomaterials in the pesticides. So they they change in response to the environmental stimulus, such as temperature, light, humidity, and pH. And so some examples of these include polymers, nanoparticles, microcapsules, and hydrogels. These are all used a lot in medicine, and their applications in agriculture are currently limited, but this is an area of current research and has lots of promise in reducing waste and the production costs in agricultural production. And these are my sources. Thank you guys for listening.